morning. If you missed our Google Meet today, this is a really quick overview of some of the most frequently asked questions that we got on our RSVP form. So first thing, the role and structure of DNA. The role of DNA is to store all of the genetic makeup and information for a cell, and it's responsible for heredity. There are three main parts to a nucleotide, which makes up the structure of the DNA molecule. So there's a phosphate group, which is this little P right here that you can see on this diagram. There's a 5-carbon sugar called deoxyribose. That's this bigger thing. It has an O with all these kind of like hexagon looking thing right here, or pentagon, sorry, not hexagon. That's your 5-carbon sugar there. And then the last part is the nitrogenous base. So here it's this C right here. And those three parts make up a nucleotide. All these nucleotides get connected with covalent bonds that hold this backbone together. And the bases, the nitrogenous bases, pair together and are held together with hydrogen bonds, which makes your backbone and then almost like the staircase looking part, and then it twists together for that double helix shape. The DNA molecule is called antiparallel. That's because the strands of DNA run in opposite directions. So you may have heard in one of the videos that you watched, they run 5' to 3', and they're going in the opposite directions, which is called antiparallel. That's how the bases are actually able to pair up. And they're held together, like I said, with those hydrogen bonds, which is just enough force to keep the bases together, but it also is pretty weak, so it allows the bases to kind of get ripped apart for replication. There are some important scientists that we want you to know about. One is Rosalind Franklin. She was the first scientist that found the spiral shape of DNA by doing an X-ray of it. Watson and Crick later found out that it's this true double helix shape with the two strands that are twisted together. And Chargaff figured out what the base pairing rules are and how much percentage of each base you should see if you take a sample of DNA. So we have 4 bases: adenine, thymine, guanine, and cytosine, A, T, G, and C. A and T are always binding with each other, so Chargaff realized they must have equal percentages of A and T in any sample of DNA, and G and C also bind with each other, so they also have equal percentages. If you look at this clip that I took from your study guide, sample 2 of DNA has 14% adenine, which must mean, because adenine binds with thymine, that there's also 14% of thymine. The same goes for cytosine. It says that there's 36% cytosine in DNA sample 2, and if there's 36% of cytosine, there must be 36% of guanine. And if you add those 4 numbers up once that row is filled in, that will add up to 100% of that DNA sample. So now that you've seen one example, if you were a little confused with this one, try out sample number 1 on your own. It's a little bit trickier because you only have guanine. All right, another really popular question was about DNA replication. So there's all these enzymes and all these steps that they go through, and this is like the really quick version of this. So helicase first comes through and unzips your 2 strands of DNA, and that makes this replication fork right here in the picture. It's able to break apart the DNA strand because of those really weak hydrogen bonds that are holding your base pairs together. Once that happens, primase, another enzyme, will come over and make this RNA primer, which is like the starting point for replication. So your RNA primer is made, and then that allows DNA polymerase, which is this big yellow thing, to come over and bind to your RNA primer, and that will actually start building new nucleotides in a chain, and it will go back and also proofread its work. So it has 2 steps. This is important because there are 2 strands of DNA where this is happening. There's the leading strand where the RNA polymerase is moving towards the replication fork, and there's the lagging strand where the RNA polymerase is moving away from the replication fork. On the leading strand, the work on replication will be continuous, but on the lagging strand, you're going to get these short little bursts of replication, and then it'll have to keep starting over and over again as the replication fork gets farther and farther away. Those little bursts, those little sections are called these Okazaki fragments. And at the end, ligase, our last enzyme, has to come over and bind these Okazaki fragments together. Actually, I forgot one more thing. This is also the reason that we call DNA replication semi-conservative, because your new DNA has one old strand and one new strand that have been binded together through the process. All right, last question was about prokaryotic and eukaryotic DNA replication. So in a prokaryote, replication happens at one point and then goes around the entire chromosome. In a eukaryote, replication happens at multiple points all at the same time, and that's the main difference between the two of those. If you have any other questions, make sure you reach out to us. Good luck on your quest, and make sure you check your answers with the study guide key. Have a great day.